So sorry, we are entering late into the predicting protein interaction pairs by arrays. So the first statement is the background is the correlation. So you take the PCA and you essentially take from the PCA whatever is correlated in the PCA you call interacting. So you have two arrays, you look at the principal components, and then you say the 50 principal components, if they are correlated above some threshold between two proteins, then you say that these two proteins interact. Okay, that's the background model. And this background model essentially leads to a simple Bayesian, not network, but a simple Bayes. And this simple Bayes, again, now I said that most don't correlate, but most associations are not interactions. But now we do a PCA, so we do a data preparation. And this data preparation is done such that we choose the PCA, the principal components that are best to predict protein interactions. So the green is the Bayes. The green is essentially the not quite raw expression data. This is a real rock type curve, meaning that random is here in gray, the diagonal. Okay, so the green is higher than the dashed gray line, and that is what you can gain by using PCA on expression data. Okay, by finding the right components in the expression data, you can sort of predict that much of protein-protein interactions. And this is ultimately, so this is clearly, the green is clearly above the gray, and this is ultimately what most publications are about. Now, when we did that here, that was the first work where anybody had really tried to machine learn to set one from the other apart. And then we get into all of these curves here are machine learned curves. And ultimately they are all the same. So what they differ in is the number of principal components the machine learning device takes as an input. Okay? It gets slightly better, so the fewer components do slightly worse. Using more does slightly better. What I do measure here is the area under the curve. Okay? What is the problem of measuring the area under the curve? So again, the curve that I have here is the false positive rate, true positive rate. And I'm saying that the method is better if it moves in that direction, if the area under the curve in that direction is higher. Okay? What's the problem in that statement? This is in fact again what you will see in most publications. So this is whenever you don't know what to do, you look up the internet and you see what others do, and this is what you would do. Why is that wrong in this particular case? So what is wrong about it? And in fact, we did the same thing here. So it's not all wrong, but there's some issue. Yeah?
Maybe because the curves with the same area under the curve might look very different. So
yes.
That it first rises and then goes, and it doesn't rise, or it just rises very late but then very steep.
Exactly. So a curve could, if that is my diagonal, a curve could look like this here and have an area none of them does. But a curve, I'm just...
Decrease again?
Yes, because ultimately, no. In this curve, not. But it could look something like that. Okay, but anyway, so it is already clear, and then so this one has a very little difference to the random, and this one has little difference to the random, right? So this is method A and this is method B. So they would say my drawing skills were substantially higher than they actually are. And say I claim that A and B both have the same AUC. Which one would you pick?
Number A, because it's better with the high.
Yes, exactly, because you can at least define some threshold where some things are right without many mistakes. And this is what, if you had an experimental collaborator and you gave them the option, well, you know, I can make you do 100,000 experiments, or I can, in 100,000 experiments, 1,000 will be right, or 10,000 will be right, or I can give you 100 proteins and one of those will be right. Most likely they will be much happier with a lower number. And if the rate will be higher, so if I gave you 50 proteins and one of them is right, so this is more this one, so the higher accuracy in this on this end, then they will be happier even. Okay, and that is exactly what typically comes out of the collaboration. What they want. In this particular case, most likely that's what they care for. So in that sense, the area under the curve here is not really establishing that. So you may argue that they are all the same because where it matters the most, they are essentially very, very similar, which is in fact the machine learning, even in this first region here, are clearly different from the raw correlation, the gray, the green, the base. So there clearly is some gain through machine learning, but the area under the curve, this region here really may not matter much. But what is clear, I believe, is that the machine learning helped. Now, this is a zoom into here, the first region, and you see that in this first region there is some overlap, but then at some point clearly the machine learning is substantially better. Okay, now when you do this SVM, you have a machine learning device that learns what is the important information in the array. Okay? So now you could ask, well, if my machine learning device learns it, why not throw the pre-analysis, the PCA, the principal component analysis, totally away and let the system learn the data directly? Okay? And this is what we show here. The red ones is you do the pre-analysis first, and the green ones is you let the system learn by itself. The green is lower than the red, meaning that the PCA helped. That still doesn't invalidate the idea that the machine learning could have learned. Maybe for a larger data set, and Michael did not revise this question because ultimately once you have enough data, this becomes less relevant as a question. But it's a sort of an interesting aside. Clearly the less data you have, pre-analysis of the data that does not do any damage to the data helps. That explains this story here. Now another way, the AUC is one way of saying that you're right. You could instead just look at, well, what's the 10% best? What's the accuracy for the 10% best, or 5% best, or 1% best? Or how many do you have in each protein in human for which you get a correct prediction? By the way, the problem with what I'm presenting here is now, so far in the lecture, we have always spoken about all organisms, or some such, all eukaryotes at least, or at least not all eukaryotes. There were different SVMs for plants and non-plants, but somehow those were very generic devices. This device here is the first time now I'm talking about something that is specific to an organism. Okay? Now all of these arrays are arrays for one organism. So you have to retrain this method for whatever organism you're interested in. We happen to have played this on human here. And again, so the paper that Michael now is sort of working on, or the manuscript that Michael is working on, is also on human. If you want to do it for mouse, can do the same thing, but you need to retrain different devices. Now, this is one way of saying that your method works. What else could you look at? By the way, there's always this issue, and let's get back to why we looked at the false positive rate and the true positive rate. The problem is, so you predict that protein A and B bind. Oh, let's begin differently. You have an experimental data set that has a set of A's that bind to a set of B's. And in that data set you have the binding between a particular protein A and a particular protein B. Now, if you predict that, fine. If you don't predict it, you miss it. It's also very clear. But what if you predicted a binding between A and C, which is not part of this experimental data set, not element of the experimental data set? What then? Is that something that is not part of the experimental data set because it's not true? Or is it not part of the experimental data set because it's not been measured? Right? How do you assess that? How are you going to assess your method? And when I look at true positive rate versus false positive rate, I escape that question. No, I don't escape that question. I still have that question. I have to still answer that question. So one way in which people address this is they say, well, I can only rely on what I have. Meaning that I can say I do not predict AB, hence I make a mistake. I cannot assess things that I predict that are not observed. Meaning I will only look how many of the ones that I know exist has my method reached. What's the problem with that one? So Gregor, what do you want?
I was just wondering if I think that there is some negative control in the experimental data. Even if you look at the protein-protein interaction database, I think there's also evidence for proteins that do not interact.
Yes, so there's
given a certain experimental method.
Dimitri Frischmann and collaborators have worked on building a data set from exactly what you talk about. So there is some experimental data, and they worked on a data set that they call the negatome. So the set of interactions that are experimentally supported, that are in fact not interacting. Okay? The problem with that one, so this is absolutely crucial for computational biology, these cases. The problem is always that because two proteins don't interact under given conditions, doesn't mean they will never interact. That is always one of the problems. The second problem with this one is it's a relatively small data set. So it's not even 10% of the positome, or whatever you call it, right? So yes, there are some, but it's still a relatively small number. But my question now back, so, okay, I take your friendly amendment, plus the negatome, okay? But mind you, the negatome is 10%. So it will be dominated by the same thing, so I take your amendment as a friendly amendment and I delete the amendment because it doesn't matter for the number in the end. But yes, you're totally right about that. In fact, they are very valuable. So in that sense, because since there is not a lot of experimental data for that, the little we have is really, really meaningful. But back to this question. So I could take the point of saying I will only see whether I predict the ones that are known, the ABs and the ACs that I predict that are not experimentally known in the negatome or positome, I will ignore. What's dangerous in this? What's the problem of that? Can you see that?
But you could just predict that all proteins are interact.
And ultimately, this is very difficult to avoid. While nobody will publish the papers who actually, hey, I have a great method that predicts every pair to interact. I have not seen anybody dare that. But there are publications, several publications, where it seems that they get fairly close to this concept. And it's just, in the way if you optimize a machine learning device, to have that as the highest value, there's just no way around it. Because you will simply only get a higher number if you predict more things. Okay? And if you have absolutely no way to check the balance, now we could take the negatome and say, I overweigh it. But now we get into new fudging of data. So this is 10% of the data, so what do I expect in nature? That there's 1 positive on 10 negatives, so I 100 times that, so I give a weight of 100, but then you compute an accuracy on something that has 100 times in front and something that has 1 in front, where you have a low level of accuracy. That's really, really, really complicated. And optimizing machine learning methods for that. Maybe there is a way, and people are trying to find a way. I'm not saying there is no way, but it's really, really complicated. Okay, that's the complication in this. The alternative is that I say, well, whatever I have observed is the complete set. Whatever is not observed is negative, and that also has many, many, many problems. But at least the mistake in that one is not going to affect that your method is going to zoom into over-predicting. The way you make mistakes here, most likely, you make relatively random mistakes. So you most likely will not quite bias the method in one particular direction. That's all you can say good about that. The other thing that you can say good about it is, essentially, it's the only thing you have today. But again, so what else, what other information could you think about? And this again, these graphs all assume that whatever is not observed is not existing. What else could we look at? How else could you assess a method? So you have a method that predicts, in this particular case, it uses microarray data to predict that protein A and B in human bind to each other through machine learning device. The base network is the background that you want to assess it against, and here I showed you one comparison of the assessment against base in more detail. So this is base, and you can do better than that. Yellow. But what else could you look at? Any idea? So you have the experimental set of proteins that interact, all the pairs. And maybe I should really write it differently. You have the set of A, B's that bind, and the set of not A, B's, the negatome. And that is ultimately the numbers we computed before. What else could you do? By the way, there's something else. I'm not sure whether I get back into that during this lecture. So typically what you will do is you will, if you train devices and assess devices, then you see to it that the experimental data set AB, so the ones that you know intact, is as good as it can be. That means you will kick out some low resolution data. Okay? When you then make the sort of inverted inference that whatever is not observed is not interacting, you could at least have an in-between level where you say anything that is supported by low level information will not end up in the I know intact, and will also not end up in the is not interacting. But it will be in the level in between where I throw away the data. So, is that clear what I'm saying? Not quite? So, what I mean is, okay, I've observed that Gary and I interact. That's by this inference, I assume that there's no other interaction in this room. And then the three of you interact and two of you interact. And there's no other interaction, okay? But then there's some low resolution information that I remember that Gary and Gregor talked, okay? This is not good enough for me to say they really interact. I cannot remember when I have seen that. But it is good enough for me to say, I'm not going to say they don't interact. So I'm going to put a sort of stop here and say, well, everybody who I'm not observing is interacting, except for those two, because there's something in the back of my head that I believe maybe they do. Okay? This is what I mean. So we have a set of everything that I know, including the negatives. And then the -1, so whatever is not observed is not interacting. And from that set, I take some out. I take everything out for which I have some low resolution evidence that they may interact. Okay? But now back to the question. How could I assess a method that predicts protein-protein interaction? Not using those. What else could I look at? Any idea?
What do you mean with using those?
So I'm trying to predict that protein A and B bind. And all my data is there. And in order to assess how well my method works, typically what I do is I use exactly what I have. And I find different ways of scoring it. AUC is one, looking at the top, blah, blah, blah is another way. There are many ways of computing the scores, but all of these scores use exactly those sets, right? Is there another way of looking at it? Is there another way of looking at the method that could help me to assess it? I'm not entirely sure whether I answer your question. Do I?
Yeah, yes, I think.
So I answered yours and everybody else is more confused than before. Great achievement. So one thing on the slide before that got us into the baby story was we have to find a way to distinguish between statistical significance and scientific significance. Scientific significance always has, let's call it a model underneath of what I believe is relevant for scientific breakthrough. And this also means that I have a problem that predicts interaction of protein in pairs. But what I'm trying to open the can is in a way where I try to see, is there another way of thinking about the problem? Yes, on a micro level I want to predict pairs. What is it? Maybe there's a macro level. Maybe there's some context in which I sort of could put these individual predictions that would help me. Yeah?
Subcellular localization.
So that is a great idea, Gregor. So you could essentially say, okay, I'm going to predict all the pairs of interactions in human, and now, since for every human protein I can predict the subcellular localization, I could revisit and ask, what's the probability of my prediction to be in the same subcellular localization? And if my method would be very bad, then between and within localization would be the same. Right? That is a clear way in which you can define the method doesn't work. Because the probability for proteins to interact between two different compartments, any two different compartments, must be different from the ones within a compartment as an entire data set. Yes, there are exceptions, but for an entire data set there must be a difference. And your method must see that. Right? If your method doesn't see that one, it hasn't learned this is the minimal thing. That is a great idea. And I'm actually not sure why we didn't do it in this particular case. I'm sure why Michael doesn't do it for the revival of this manuscript, because he's actually using that information. And this is partially why you work on the subcellular localization, because he needs that. That's a great idea. It's exactly the kind of thing I mean. Something else. Like that, yeah?
You can also look at how these interactions that you predict cluster. So if you have only very sort of only 2 proteins and they're sort of isolated from other proteins, that's sort of unlikely, and maybe you would have things like that.
And isolated you mean by what? By family? What do you mean by cluster?
Isolated, I mean by not interacting with each other. So if you only have small clusters that are completely disconnected, then it's sort of strange, because
yes. So we're going to the next level. We're actually ultimately we are saying that we need the pairs to build up networks. So let's just look at what network features we have. So let's just look, you looked at one feature. Am I going to have hubs or something? Or am I going to have closely connected cliques or something like that, right? But whatever feature we look at, here what I'm showing is network distance. You could look at a bunch of typical network features and ask, are the predictions resembling what we know the natural networks look like, right? This is sort of a similar direction. You simply ask, put on the level of a network, do you at least get some of the features that you expect? And what we see in the, here we show the SVM score. So more reliable scores are here in that direction, and that's true for base 2, and that is overlaid to the network distance. So for the base, so that is the green curve that I showed you, essentially is taking the microarray data directly to infer that 2 proteins interact. And when you do that, you see that you have essentially a completely flat distribution, meaning that no matter what the base score is and no matter what the network distance is, there is no difference. While in the SVM approach, you have the effect that things that are closer in the network, the cluster, are predicted with a higher reliability. There's a tendency for this. And this is clearly not built in, because this is a network feature, and in this training here, the network feature was never learned. This is an example where that is what you want, this is what you don't want. This is random, because there must be a relation. Things that are closer in the network, they must be stronger in your prediction method, right? And then, you know, you can look at a bunch of other features. You can look at other data, synthetic rescue, phenotypic enhancement, biochemical activity, and see whether there's sort of an enrichment in some sense. So you have a set of things that you, with a high reliability, predict to interact, and now you look at data, a little bit like the localization story, where you're not quite sure that this synthetic rescue is really directly telling of protein-protein interaction. It's not, but you want an enrichment under this. And so you can test various environments, so this is not on the network level, it's on a phenotypic level, it's on the level of the organism. Do you at least capture some other effects? And you can sort of play around with this and introduce a couple of other ones. Then you can, another thing that you should actually look at is, are there some of your predictions that you really trust? Is there a level, in this particular case here, the most trusted, I can't remember what the accuracy was, but the accuracy even of the most reliable ones was still pretty low. I believe the way I remember it, Tatsing, so I assume that this one here was done on he analyzed 100 papers or something like that, or 100 proteins for which he read all the papers, and then he came up with these kind of numbers. This is a tiny subset of what you do, but the tiny subset is chosen by the most reliable ones, and then you sort of have some evidence, meaning that you want to sort of have some evidence that some of what you predict and you're most reliable has meaning. And that is a sort of complicated issue. Here are some evidence of things. Now it gets really complicated when you get into things where essentially you predict something that is new. So now the definition of new is nobody else has seen it before. And now you want to see in the literature that it could be true. Nobody has ever seen it before. Show me the paper that says it could be, right? And that is sort of a big guessing game that you have to be very careful how to play. Because it's very easy to read things into descriptions, but but this is what you're left with at that point. Now, ultimately, at the end of the day, you clearly can predict physical interactions, transient interactions from microarray data if you train machine learning devices. Now, and I will have to stop here, I'm afraid. Slide 58. So I will not go through the 248 on Thursday. I will do whatever subset I can do. And this part is about prediction. Clearly is the acid test for understanding. But what about machine learning? So that brings us back to the question of the points that I showed you. They differ between different interfaces. Does it mean that I can actually predict? In that particular case I showed, I can predict. Now, does prediction, and that prediction was based on machine learning, does that imply that I understand? And typically the answer to this thing is sort of done in some sense like this. Maybe people don't use the medieval or the sort of simplistic image, but ultimately, in my view, from where I'm standing, the whole world of biologists and also other computer scientists has the vision of this, or has this image of the black box. Machine learning is some black box, and the black box sort of is a sink of understanding, and clearly machine learning is not understanding. And that is a point I want to repeat on Thursday. And I'm trying to sort of, I believe, say that this is a misunderstanding. Thanks for your attention. See you on Thursday.